13th, okay, Friday the 30th here. We didn't pick this on purpose for Reza, okay? It's a pleasure for me to introduce Professor Reza Ovissi. He is originally from Iran, and he actually finished his master and PhD from Tarbiyat Modares University in Tehran. This is a very prestigious university there that has graduate programs, actually. He graduated with a PhD in 2010, and then joined Washington State University at the rank of research associate. And, and there was the manager, the research and development manager at Stolt Sea Farm from 2015 to 2016. And a postdoctoral researcher after that at UC Davis. In the middle of this adventure, he actually got a PhD from Washington State University in biological systems engineering. His traditional or his original work was mainly focused on seafood science and technology. So he actually combines these fields on his research. He does a lot of extension. And I think it's an opportunity for us all to see a little bit more of the professionals that really do work outside the lab and on the extension part of Virginia Tech. So without any further ado, I'm just going to introduce Reza. Thanks a lot for accepting the invitation from being here today, and I'm really looking forward to your presentation.
Thank you so much, Carla, for your introduction, and thank you so much for inviting me. It's a great pleasure for me to be here. So my name is Reza Ovissi. I am assistant professor at the Department of Food Science and Technology and the Seafood Agricultural Research and Extension Center. My lab is located in Hampton. We have a research facility here working on different topics. My program is Sustainable Food Production Systems. And today I will be talking about one of the technologies that we have been working on that since 2018 when I started my job here at Virginia Tech. And it is related to nanogas or nanobubbles. And we have been trying to use that and optimize that for different applications in food supply chain, including microbial food safety concerns. In next couple of slides, I would like to kind of bring some introduction about my program here and introduce you what we are trying to do here. And then after that, I will be focusing on the nanobubbles technology. So my program here at Virginia Tech is Sustainable Food Production System. Because of the diverse background that I have, I've been always having problem with explaining my program to people and what I'm exactly doing here. So then I decided to kind of focus on one topic, sustainable food production systems, which makes my life easier. And under this umbrella, I can work on different topics. I'm mainly focusing on food safety and quality, alternative proteins and bioprocessing, and cell culture and tissue engineering. Before going to these examples, it is important to note that, like Carla said, my program here is 50% research and 50% extension. I don't have any teaching appointment, but I have so many postdocs and students, and I am collaborating with industry, but the nature of the work that I have with industry is pretty diverse. For example, I provide very simple service for them. Sometimes they call us and we go to this food processing plant. We take samples from the drain, from the contact surfaces. We bring them to the lab to make sure that there is no pathogenic bacteria in the environment after sanitation. Or we provide some fact sheets, very simple and lay language for them. So sometimes we are working with some big companies on, for example, developing new packaging material. And my main focus, probably more than 90% of my effort for the extension, is mainly focusing on working with the startup companies. And I'm working with so many startup companies, and some of them, they are not a startup anymore, and they are technically large companies. And we are working with different technologies and different methods for producing foods, and technically any method related to sustainable food production systems. Well, giving you some example, I would like to give you some examples about some of our research in the lab related to food safety and quality. One of the systems that we are working with, one of the animal production systems or food production systems, is Aquaponics. Technically, it is a joint system for fish production in a RAS or recirculating aquaculture system with plant material. And you can see here a picture of the kind of a small system that we have. In these tanks, we keep fish, and the water from the fish, they are going through these plant parts, and they are providing enough nutrients for the plant, and they grow. But the main or the heart of the system is the biofilter, and the biofilter is a kind of, we call it K1 plastic materials. They are pretty small beads or plastic beads, and we create good bacteria consortium or microbial biofilm on them, and they are taking care of the quality of the water. They are improving the water quality. And not only the water quality, in many cases, if we have good established bacterial consortium, they can reduce the pathogenic or opportunistic pathogenic bacteria in the system, because the ultimate goal is reducing the antibiotic application in these systems. So last year, we were able to develop a reactor, 500 liters, and we challenged this reactor after developing the biofilms for 15 days and maturing them. So we challenged that with Vibrio parahemolyticus, and we were able to get rid of the Vibrio with 4 log reduction within 24 hours without adding any chemicals or antibiotics into the system. And this year, we are establishing these reactors in industry for cleaning the water before introducing the water into their system. Talking about antibiotics, I have another project. We are trying to understand the antibiotic-resistant bacteria prevalence in oyster in Bay Area and also in water. We have been working with Virginia Department of Health for collecting the oyster. We are almost done with this project. As of today, actually, my students and postdoc, they finished the project, and I haven't seen the results, but so we are working on that at this moment. And also meanwhile, we are trying to develop some fact sheets for people, because we did a survey study, and for public, we collected some information from public and see where is the gap exactly regarding the antibiotics application at home. And we are trying to develop some fact sheets based on that. I have so many collaborators with extension and consumer service background, and they are helping us to develop these fact sheets to train the people. And so we have other projects like photosensitizers. We applied plant-based material, activate them by using light, for example, LED and UVA, and they kill the bacteria easily in water surfaces on the surface of the food material. And also nanobubbles, which I'm going to address that today as one of the examples. Recently, my lab also received $1 million from USDA to address COVID-19 in food supply chain by understanding the virus survival and how this virus can be transferred through the food supply chain, and how we can develop some mitigation strategy for killing the virus. We are working with live virus isolated from people, and in a BSL-3 lab under Dr. Andrea Bertkus supervision. She's one of the PIs of this project at Virginia Tech, and we have two groups of team. Technically research group and also extension group. We are working with stakeholders, and we are trying to translate the results that we are collecting from the research part into a lay language and useful information for industry. So I think within the next couple of months, you will hear a lot about this project, and you will see some part of the results available sometimes in late, probably January. Another topic that I've been working on is related to alternative proteins. I am working with insect protein, algae waste, and plant material. We have several funded projects to develop plant-based diets for both human and animal nutrition, and insects for both human and aquafeed development. For example, here you can see some of these insects, dried one, definitely. So we extracted functional protein from them and the oil, and we are using them for different purposes. One of the publications is under review at this moment regarding this topic, and we are working with different stakeholders, and we just awarded a grant in collaborating with other researchers at Virginia Tech to develop aqua feed from insects like black soldier fly and protein hydrolysis from this black soldier fly. I'm also working on cell culture and tissue engineering, and if you have any question regarding these topics, and if you are interested to learn more about that, I will be more than happy to provide information for you. We just set up our reactor in the lab, and yesterday we set up our inverted microscope. And so we have everything in the lab for moving forward on these topics. So with this, I would like to go through the nanobubbles technology. But before talking about this technology, let's see why food safety is important. So I brought some information here for you to understand the burden of the foodborne pathogens in global level and in the US. Well, I took this from the World Health Organization. And as you can see, based on the report that they provided, 1 in 10 people, they get foodborne illnesses every year. It's a lot, it's a big number. And 420,000 deaths we have annually all around the world, and 1/3 of that, they are children. So it shows that these numbers are important, and we have problem with the food safety and also foodborne illnesses. But let's take a look and see what are the problems in the United States. Well, in the US, 1 in 6 Americans, they get sick annually because of the foodborne pathogens. We have 128,000 hospitalization and 300 deaths. I brought an example here for you as one of the food commodity. For example, for seafood, because I've been mainly working with seafood, from 1998 to 2015, we had more than 200 outbreaks related to seafood products and more than 4,500 bacterial and viral illnesses. The rest of that, they might be related to allergens, and which is not related to our topic today, but those that they are related to microorganisms, here are the numbers. And technically it shows that how important it is, and how we need to actually control them. And let's take a look and see what are the sources of these microorganisms in the food supply chain. The most important problem that we have in food industry is coming from the biofilms. And as you know more than better than me, so biofilms created by bacteria, right? So more than 90% of the human infections, they are related to biofilms, microbial biofilms. And more than 65% of foodborne illnesses, they are also related. They can be traced back to the biofilms. So when we are talking about the biofilm, so we are talking about the high organic loads because they have high amount of the organic loads. As a food scientist, if you tell me you have high organic loads in your plant, the first thing that comes to my mind is then you have to apply high amounts of sanitizers at high concentration. They have EPS, biofilms, they create EPS and they protect the bacteria and they prevent sanitizers to become in contact with pathogenic bacteria, and they increase the tolerance. So we have to apply high amount of sanitizers. We need to apply mechanical force, and sometimes the regular sanitizers, they don't show any efficacy. So here is an example. I took this picture from a plant, and you can see how they apply sanitizers in the foam and high concentration, and they leave them there. So they have problem with, most often they are corrosive and they are not really healthy material for, they are really concerned about the employees' health as well. So we have been taking samples from different places. This is a food contact surface, as you can see, to make sure that there is no bacteria or biofilm, because they can easily form biofilm, and also from drains. So technically removing the biofilm is not very easy in food industry, and we need to actually develop some other technologies for controlling them. So, but the other problem that we have in terms of the sanitation is just the food material by itself. So assume that you have a spinach leaf, right? So you have surface roughness, and because of that roughness, the bacteria easily can hide themselves into those roughness, and they can actually be protected when we expose them to sanitizers. We have organic loads on the surface of the food material, and the surface is very complex. So organic loads will consume some of these sanitizers, and you have to add at least 200 ppm when you are washing lots of fresh produce to maybe get around 10 to 15 free available chlorine that can kill bacteria. So regular sanitizers, they have limited efficacy, and usually we are talking about 1 to 2 log reduction after applying that much effort. So these are the problems that we have. For example, if you have a surface like this, you have roughness and cracks, and bacteria, they can easily go there and stay there without being in contact with microbubbles, because microbubble, that's one of the major issues during the washing steps. And this is a SCM picture of a part of the plant. And as you can see, bacteria, they can easily hide themselves inside of the cracks and the pores on the plants, and sanitizers, they cannot really go there. So not only we are concerned about the microbial food safety, but also we are concerned about the microbial spoilage as well. I brought some numbers here. I don't want to really go deep, but I just wanted to show you how important spoilage is. Spoilage technically is happening just because of two main reasons. Chemical spoilage, which we cannot really control that, and the only control for that is temperature control, and microbial spoilage. But we can control microbial spoilage pretty easily. If you can see here, so more than 20% of the meat, dairy, and oilseed is being wasted globally, 35% of the seafood, and close to 50% of fresh produce. In the US, over 45% of fresh produce is lost because of this spoilage. So it shows that by considering this fact that 30% of our energy is being used for food production and more than 80% of our fresh water is also being used for food production. So we need to make sure that we are using our resources better, and we need to apply different technologies to increase the shelf life of the product and reduce the risks of food safety or foodborne pathogens. And by that, we can reduce the waste. So technically, the only way that we have is enhancing microbial inactivation by applying different technologies. Let's take a look and see what is the current practices in industry. There are different types of chemicals. I brought some of them here, and we call them conventional sanitizers for food industry. People, they have been using quats, peracetic acid, sodium hypochlorite, hydrogen peroxide, and recently electrolyzed water or neutral electrolyzed water, which is called NEU, or the main component is hypochlorous acid, is not emerging anymore. It has been used by so many companies and people, they started using them. And these are kind of the conventional methods, but they have problems. So we have still problem because we need to apply high amount of these sanitizers. They have limited efficacy. Still they have organic loads on the surface of this food material. Surface roughness is another issue. And sometimes chemical handling is kind of hassle for food industry. So we need to find another way to use or to deliver the sanitizers or remove the bacteria from the surfaces, bring them to the cell suspension, and then by less amount of sanitizers, we can easily kill them. That's why we started using nanobubbles technology. Excuse me. So nanobubble has different name. Nanobubbles, nanogas, or ultrafine bubbles. Some people, they use ultrafine, even though according to this graph, ultrafine is kind of larger. But we are talking about bubbles around less than 200 nanometer. And when we are talking about something that small, they have so much functional properties. They have negative charge, and they are pretty strong. They have so much energy inside of them. The pressure, internal pressure, is close to 6 megapascal, and which means if we burst them, we can release so much energy. And if we release so much energy, we can theoretically, so we can convert oxygen to reactive oxygen species, and we can release more hydroxyl radicals, which can kill the bacteria. They reduce the surface tension, and so they have Brownian motion, means they can stay for a long time in the water without having any problem with bursting them. I personally actually created some nanobubbles with pure oxygen in the lab, and I measured the oxygen. It was more than 40 ppm. Regular saturated level is around 9 to 10 or 12 maximum, but it was 40 ppm. After 3 days, I went back to the lab. I measured that. Still, it was 15 ppm. It's pretty high. It's pretty stable in the system. And also the good thing about that is you can use different gases for that. Here, as you can see, we have different types of applications. For example, water treatment, environmental treatment, or hydroponics, food industry, and other type of industry, but it needs to be optimized for any other purposes. And also we found that, so if we apply this nanobubble, let's say oxygen nanobubble at certain level, so we can actually boost the bacterial growth in the system, particularly for those systems that relying on microbial community. If we optimize that system and apply and the concentration of nanobubbles, instead of killing the bacteria or removing them, we can enhance the system. And still we are working on that to optimize that. They can penetrate into the materials, including biofilms, and technically this has been used in petroleum industry for removing the organic loads. Here is another example, putting them together, and as you can see, the smaller bubbles that we have, the better properties that they provide for us. So the first things that we did in our lab, we wanted to see what is the impact of the gas source on antimicrobial properties. So what we did, we created some nanobubbles with oxygen and carbon dioxide. And so we exposed them bacteria, in this case, Listeria innocua. So we put the Listeria innocua on the surface of a spinach, and we had 10 ppm sodium hypochlorite as our available chlorine. And we exposed these to different solutions. So deionized water didn't reduce that much of the bacteria. Same thing with the CO2 nanobubble. Oxygenated nanobubble kind of reduced a little bit of the bacteria from the surface. Again, it's not killing, it is just removing. And when we had deionized water plus chlorine, we had a little bit reduction on the surface of the spinach. But when we had CO2 nanobubble plus chlorine, we didn't show any reduction. It didn't work perfectly. But when we had oxygenated nanobubble plus chlorine, we had more than 2.5 log reduction on the surface of the spinach, which is showing that the gas source has important role on removing or killing the bacteria. So in next step, we decided to use ultrasonication and combine this ultrasonication with nanobubbles technology and in cell suspension. What we had was just a cell suspension, no other organic loads. And what we found was, for example, when we had E. coli and exposed the E. coli in cell suspension to nanobubble and ultrasound for 15 minutes, so when we had our control group positive and negative, ultrasound and nanobubble, we didn't see that much reduction. So technically no significant differences. But when we apply nanobubble plus ultrasound, we had more than 3 log, almost 2.5 log reduction after 5 minutes for E. coli, and after 10 minutes and 15 minutes, they were all gone. So we were not able to detect E. coli after applying that. And same thing with Listeria innocua, but Listeria was a little bit more resistant. We didn't see a complete reduction. And this is very important to note that. So we also run some other experiments. We conducted some experiments and we found out, so the ratio between the number of the nanobubbles and the number of the bacteria is really important. If we have more bacteria in the system, technically nanobubbles cannot really provide that much efficacy for us. So we tested from 10^4 to 10^8, and 10^8 was not really actually pleasant. The results, they are not really satisfying. So, and it shows that it needs to be optimized, and it's based on the number of the bacteria as well. So in another study, we started working with some of these pathogenic bacteria in aquaculture. And this is our aquaponics system. As you can see, we have basil and spinach here, and this is our fish tank. I don't know if you can see the small fish here. And so we took the water from here without any filtration. We had all the organic material inside of that. And then what we did was we inoculated the system with some pathogenic bacteria like Aeromonas hydrophila and Vibrio paramelliticus. And we exposed them to ultrasound and nanobubble. And as you can see here, for Vibrio paramelliticus, we had, after 15 minutes, we had 2 log, 3 log reduction. But for Aeromonas hydrophila, we had complete reduction after 10 minutes and 15 minutes. It shows that this technology, after optimizing, we can use that for treating the water in aquaponics, aquaculture, and even hydroponics system. And also it could be used for water treatment in, water treatment industry. So we also applied this technology for the ultrasound and nanobubbles technology, technically combining them. So on spinach. So we wanted to see how effective this combination is when we apply that for attached bacteria. That's the main concern on the surfaces. Well, just forget about the quality of the product. The quality of the spinach after 20 minutes exposure to ultrasound was not really good, but we just wanted to see what is exactly happening for the bacteria on the surface. So this is the ultrasonication. We had 1 log reduction that was not really good, but when we combined that with ultrasound and nanobubble, we almost had, I would say, 4 or 5 log reduction. Listeria inoqua was a little bit more resistant on the surface, which is one of the things that we were expecting that. After this, we tried to combine this technology with surfactants, but what happened was cationic and anionic and neutral surfactant, they didn't really work, at least in our preliminary tests. I didn't bring the results because we did that once, and we need to actually go deep and understand that better. So technically what we are trying to do is we are trying to see how we can combine this with other technologies and reduce the chemical application, right? And in this case, it didn't really work. I mean, nanobubble by itself was able to remove bacteria better than combining with surfactant. Technically, it can show that nanobubble has some surfactant property, but we need to actually work more on that and make sure about that. Well, so we did another thing. We wanted to see that because there is a kind of argument that people, they believe that nanobubbles, they don't exist in bulk. They attach to the surfaces. We thought that if we expose the spinach, inoculated the spinach to nanobubble for a certain amount of time, and we give them some time that they can attach to the surface of the spinach, then we will be able to get better results. We were able to kind of incubate them with nanobubble and then expose them to ultrasound, and we got actually a little bit better results about that. But this is not the results that we are trying to publish. That's the 1-time thing or 2 times experiment. But I just wanted to share that with you, that we have been looking at these combinations from different angles, and we have been trying to answer at least our own questions, scientific questions and research questions. So another thing was we wanted to see that how nanobubble treated samples or surfaces can prevent bacterial attachment. Well, so we treated some plastics and a stainless steel, and then with nanobubble for 10 minutes, and then we dipped them into the solution, PBS with bacteria, no organic load, just with bacteria, and in this case, just Listeria inoqua. And then after different certain time, we went in and grabbed them and measured the bacteria on the surface of them. What we found was, so as you can see here, it's not big difference, but still it is interesting that how nanobubble treated samples, they reduce the bacterial adhesion. And then we measured the contact angle on the surface of these non-treated and treated, nanobubble treated samples. And we see that on plastic and stainless steel, when we treat them with nanobubble, the contact angle reduces. Maybe not the huge reduction, but a little bit reduction. We can see that here. So, but let's go back to this synergistic impact. What we did here was we tried to actually understand what's going to happen for nanobubble in combination with chemical sanitizers. We tried electrolyzed water in this case because we wanted to have everything kind of emerging, something like that. So what happened was we had 3 types of bacteria, inoqua, E. coli, and Vibrio paramelliticus. We developed some biofilms on plastic and stainless steel, and we exposed them to 4 treatments: control, nanobubble by itself, 10 ppm neutral electrolyzed water, and combination of this electrolyzed water and nanobubble, and for 2, 5, and 10 minutes with and without 200 RPM. And we found that shear force or mechanical force is really important. So we did that once without shear force, and then all of the experiments from that moment on, it was with shear force. And other people also proved that shear force has an important role in removing biofilms. So Vibrio was the most sensitive one. After, I would say 2 minutes, we were able to kill everything on plastic and stainless steel with electrolyzed water and the combination of electrolyzed water and nanobubble. And even after 5 minutes on both coupons, we were able to remove or kill, in this case, better say removing biofilms even with nanobubble. So it was quite interesting that nanobubble by itself was able to remove the Vibrio. So let's take a look to the E. coli. So E. coli was more resistant. And as you can see here, we had reduction with nanobubble, but not complete reduction. But in combination of nanobubble in combination with electrolyzed water was able to kill the bacteria perfectly. So, and on a stainless steel, it was a little bit more resistant. So, but for Listeria was quite interesting because when we had Listeria innocua on a stainless steel, we can see the trend here because that's a little bit more resistant. So nanobubble was able to reduce within 15 minutes. Electrolyzed water, same thing. But the moment that we combine electrolyzed water and nanobubble, the efficacy of electrolyzed water was improved compared to the other treatments. That's why for Raman confocal macroscopy study, we focus on the Listeria innocua biofilm on a stainless steel. We couldn't use the plastic one because of the interfering that it's gonna provide with the laser. So we had to use a stainless steel, and also technically it's not Raman, it's a SERS, surface enhanced Raman spectroscopy. We applied silver nanoparticles to boost the spectra and scattering and be able to collect them. And in next slide, I will show you some picture. This is the chemical picture. So technically what's going to happen is Raman is going to provide this chemical picture. And this is the control group is after exposing the biofilm on a stainless steel to just PBS. It's the control group, right? And as you can see, the area that we have color, they have biofilm. Different colors mean different structural things in terms of the chemistry. And the white areas, as you can see, is the plain area. We don't have anything on them. So it's just no organic loads. And after applying nanobubble by itself, we had so much reduction, right? It's very interesting. And electrolyzed water, same thing. And in combination, it was reduced, but a little bit, not really, I would say not really significantly. So we had the spectra from the Raman for different area, for example, for amide 1, amide 2, and also for EPS polysaccharide, for phenylalanine, which is indicating the protein and DNA area, and again, deformation of the EPS. And the band intensity, the peak intensity, they were reducing by going through these treatments, and which is interesting. So in next slide, I would like to show you their maps from this Listeria innocua biofilm on a stainless steel exposed to different solutions. We have different peaks area. For example, for this one, it's related to the deformation of EPS. This is the control group, not that much changes. And the more red color that we have, more dense material, at least for that particular peak or organic material or a molecule that we have. And the less red material means, so we are technically biofilm is being disrupted. So by going from control to nanobubble and nanobubble plus electrolyzed water, as you can see, all of these red areas, they are becoming more blue, and we are losing our biofilm, and biofilm is already being distracted and disrupted. And it shows that the nanobubble by itself also can have some interaction with protein, polysaccharide, and also the DNA. But further studies are required to go deep and extract these different materials and study them. So we haven't done that so far. This is a conclusion that we have. What we learned from all of these experiments that we conducted, nanobubble can significantly remove microbial biofilms on different surfaces. And they can be burst by ultrasound and create some ROS and radicals and kill the bacteria. Efficacy will be improved if we combine these technologies together. And shear force has an important role. We should actually consider that. Nanobubble can induce microbial injury, which is really interesting and important. And so by using nanobubble, we can reduce the antibiotics application or sanitizers application. And we can actually engineer the surface to deliver the materials to bacteria. And in many cases, they can easily do that. They can deliver the material to the bacteria. They can penetrate into the biofilms and organic loads. We can use them for water treatment, agricultural water treatment, and removing biofilms from pipes. That's one of the things that we have been discussing this with many companies. They are very interested in this. And membrane sanitation, or cleaning in food industry is very important, and it could be used for that purpose. Wound healing is not related to food industry, but wound healing is one of the things that we can think of, and we can use this technology for wound healing as well, or removing the biofilm from the wound material. So I just wanted to introduce my team at the Virginia Tech. Dr. Dusher, she's a postdoc working on tissue engineering. Dr. Janak Dakhal, he recently joined us for the COVID. He will be working in the BSL-3 lab on campus. Dr. Kramat and Miaya, so they are both working on alternative proteins and aquafeed development. Shamil, he was my intern, and he was a really great researcher, and he went back to his country. Right now he's applying for PhD. And technically, he was working hard on nanobubbles technology, and he tried to optimize that. And Ayat Batish, my PhD student, she's working on tissue engineering, tissue engineering, and Julia Kesselling, so she's working with me and another faculty, Dr. David Kuhn, on gut biome. And we are recruiting another PhD for the next fall to work on the gut microbial community, on insects fed fish. Also, I would like to appreciate my collaborators, Dr. Setar Shirudi, who was working with us and initiated this technology and work. She's working for VDAX right now. Dr. Michael Schwartz, director of the Seafood AREC, and Dr. Nitin, my postdoc supervisor and my mentor during my postdoc. I have been working with him. I was lucky that I have him as my mentor. Still, I'm working with him, and also the funding agencies, Sea Grant, USDA, NIFA, Hatch Project, and Virginia College of Agriculture and Life Sciences. And we also have some of our papers available, one of them published, one of them under review, and several other papers, they are under preparation at this moment. And with this, I can take questions. Thank you so much.
Thanks a lot, Reza. We have a couple of questions from the audience. I'm gonna kick off with one question of myself, if you don't mind. I would like to know for how long the nanobubbles treatment protects the surface from biofilm formation.
So how long they can prevent, or how long it takes for them to remove them?
Prevent.
Oh yeah, so that's something that we are trying to understand that at this moment. So probably nanobubble by itself, based on my own experience with them, nanobubble, they cannot really prevent 100% if we just use them once. If we use them constantly, probably 2 times per day for washing a specific, surface, then they can prevent that pretty easily because they can stay on the surface, right? But if we don't apply them, just apply them today and a week from now come back and apply them, that's not going to happen. They cannot really prevent that unless we mix them with other type of polymers.
I see.
So if we mix them with other polymers, my hypothesis is they can stay there for longer time.
Sure. In fact, a follow-up question on that comes from Pat from the audience. She asked whether you have tried to use ozone, oxygen-3 in the nanobubbles. Just fill the nanobubbles with ozone. I guess that's what you meant.
Yes, that's a great actually point, and we are working on that, and that's our next step. We haven't done this so far, but that's our next step. And technically, if you have ozone and nanobubble, so technically you will have better efficacy. And so that's another interesting topic that we have been trying to answer that, yeah.
You might get more free radicals or something.
Absolutely, yes.
Okay, there's another question. How does stimulated nanobubbles compare in efficacy to current practices for sanitation?
So if we are talking about, the measure method is just the log reduction. So in our research, we showed that when we combine, for example, nanobubble with the current practice, which is just the regular washing system, we will have at least 2 more log reduction. Instead of having 1 to 2 log reduction, we will have 3 to 5 log reduction on the surface, if we combine that and the right concentration with right sanitizer.
Sorry, I was mute. There's a question that relates to a clarification about how you run an experiment. You measure the survival rate of bacteria in spinach. How do you design the experiment? Do you measure the bacteria in the water or the one on the spinach?
So yeah, that's a great point. So no, actually, so when we have the nanobubble, so we need to consider this. We need to be very careful about that. Nanobubble, sometimes it doesn't kill the bacteria. So in many cases, it just removes the bacteria. In this case, we need to measure that on the surface of the spinach and also in the water as the leftover. So for the ultrasonication, we did that and we showed that there is not that much bacteria in the water. So technically, ultrasonication was able to remove and kill. But for some cases, for example, for nanobubble plus biofilm, it is just removing. It doesn't kill them. So it removes them. So for spinach, it was on the surface and in the water. But the results that you saw, it was just the surface of the spinach.
Okay, we have another one. Does the raws, the radicals, pose any risk for human consumption of the food after this treatment is completed?
No, so nanobubble by themselves, they don't show any risk. And as a matter of fact, it was invented by Japanese researchers many years ago, and people, they have been drinking that. And there is one application for nanobubbles for beverage industry. And so it doesn't show any risks for drinking nanobubble. I mean, as far as we are at this moment, nothing has been proved against that. Yeah.
Yeah, well, nanobubbles are used for therapeutics as well.
Right, exactly.
We have another question. Do you vary the frequency for sonication? And if so, do you observe different microbial reduction? So
there is, high frequency and low frequency has been used. I just used one method because my sonication machine doesn't provide two types of frequencies. I have seen other people, they were working, and based on my own experience, one is working better than the other one. I think we used a high frequency and not really low frequency, if I remember that correctly. Yeah. So we just tried one of them.
We have one more question. Can we use in SIP system in plant industrial?
Seed system?
SIP. I don't know what SIP is. CIP. I'm not familiar with that as a question.
I'm not familiar either.
Could you please, the person that asked that question, clarified on the box for me? Clean in place.
Oh, yeah. Clean in place. Yes, absolutely. So that's the goal. And so it needs to be optimized, right? It needs to be optimized. And that's one of the discussion that we had with industry, because they were saying, so we need to purchase the reactor, right? The reactor is not that much expensive though, but because the quality of the product that you get from using this machine is really good. And so it needs to be optimized for that purpose. If you have aquaponics system, you don't need that much nanobubbles, but if you want to sanitize a big plant, you need so much nanobubbles. So, but yes, it could be used for cleaning plant, cleaning place purposes. Yeah, that's the goal actually.
Have you tried water saturated with oxygen and compare it with nanobubbles?
Water saturated with oxygen.
Mm-hmm.
So water saturated with oxygen is just not stable. It's just saturation level in water with regular bubble is just around 10 ppm.
Okay.
Technically, it's not really good comparison.
And there's a pushy from the audience. They want to know when you're gonna get data with ozone and nanobubbles, and if that is a paper that is in the works, if you have any idea when it's gonna get published. And now you ignite people's seek for knowledge, and now they want to see your publications. So you need to tell us when this is going to be available.
Sure. Going back to the last question is, we tried this not in terms of the oxygen. We tried this with the regular, I would say, washing system in industry, because oxygen is not our concern in food industry, right? So it's just the way that we wash them, and we call it microbubble. So in that, during that washing system, we just use a microbubble. And so we tried microbubble and macrobubble, and compared to those, yes, nanobubble was more effective.
Okay. Any other questions for Reza? There's a lot of questions. Okay, well, thank you very much for joining us. I had to admit that, oh, there's one more question. Do you mind?
Yeah, go ahead, please.
Okay. Do you think any other oxidizers would work well with nanobubbles? Do you have any idea which ones?
Sure.
Have you tried more than one oxidizer together? I mean, I think the question is very pertinent.
No, it's okay. Yeah, that's perfect. So yes, we are trying to see, we applied that with hydrogen peroxide and peracetic acid. So because these are the conventional method, and people, they have been using that in industry. And so, but let me explain this in this way. So when you're talking about these combinations, it's not just the combinations of nanobubble with these technologies, right? It's a combination of these with against what bacterium. So, and under what condition? Is that the cell suspension, organic loads involved? We need to optimize that, and we haven't done all of this, and I cannot really provide which one. But based on my experience working with CO2 and O2 was better compared to CO2 with chlorine-based sanitizers.
Sorry. Thank you very much for joining us today, and I'll see you next Friday. Thanks a lot, Reza.
Thank you. Thank you so much.
Bye.
Bye-bye. Have a great day.
You too.